The Citric Acid Cycle by Laureano Andrade Vicente and the MCAT Disciples. Here are the AMC topic list. As you can see, it's quite short, but we have a lot to cover. So let's get right into it. Okay, so a quick review from last time. We talked about the straight chain carbohydrates. I showed you the example of D-glucose and L-glucose. So these guys are known as enantiomers. They differ at every single chiral center available to them. The designations D and L refer to the stereochemical. It designates their stereochem. And we look at the second to last carbon. Okay, it's numbered 1, 2, 3, 4, 5. We look at this carbon right here, and if the hydroxyl group is on the right, then it's D-glucose. If it's on the left, then it's L-glucose. How we name straight chain carbohydrates is as follows. This is a 1, 2, 3, 4, 5, 6 carbon straight chain carbohydrate. So it's going, and it's an aldehyde functional group, so it's going to be called an aldohexose. Okay, that's how you name that. Alright, and then we can close that ring by doing a nucleophilic attack on the carbonyl carbon. In this case, it's going to be an aldehyde, so it's going to form this hemiacetal functional group. It's going to form this closed heterocycle. And we can get the designation alpha or beta when we do this ring closure because of the fact that this nucleophile is attacking a trigonal planar geometry molecule. Okay, so then this can end up going up or down. This OH here that's going to form after this guy attacks can go up or down. When it's going up, it's called the beta. So it's called the beta anomer, okay? Because we're looking at the anomeric carbon. And if it's going up, it's going to be called beta. If it's going down, it's going to be called alpha. And up or down is in relationship to the hydrogen that's also bound to this molecule at this position. It's had nothing to do with position 6. Position 6 in a closed form is going to tell you whether it was a D-glucose or an L-glucose. If it's going up on position 6, it's D-glucose. If it's going down, it's L-glucose. Okay. We talked about how to convert Fisher projections into the Haworth projection, which is one of these guys. So if the group is on the right, then it's going down in the Haworth, and if it's on the left, then it's going up. That doesn't count for things connected to the endocyclic molecule, so oxygen. All right, and again, you notice that we can call this alpha D-glucose or alpha D-glucopyranose. The pyranose is a 6-membered heterocycle. That's how you name a 6-membered heterocycle containing oxygen. If you had fructose here, you can call it alpha D-fructose, or you can call it alpha D-fructofuranose. That's the same thing. The furanose is just a 5-membered heterocycle containing oxygen. We also talked about high energy molecules. These are the molecules that you can couple to, let's say, the formation of ATP to then form ATP because it has a negative Gibbs free energy associated with this. The breakdown of these products, of these compounds, is associated with a release in energy. And so you can use that to power reactions, which you'll see in citric acid cycle as well as you already saw in glycolysis. And then we'll just review glycolysis very quickly. We start out with glucose, right? This is all about carbohydrates, breaking down glucose into then molecules like ATP. So glucose via hexokinase can form glucose 6-phosphate. Hexokinase uses the proximity and orientation catalysis mechanism and the magnesium ion, and it uses an ATP to get that phosphate onto the glucose. Of course, it has magnesium ion. It's using ATP. Glucose 6-phosphate, the fructose 6-phosphate via phosphoglucoisomerase. It is a ring opening closure mechanism. It's an isomerase. It gets free energy is about 0. It's relatively easy to do so. Ring opens, ring closes. Okay. Fructose 6-phosphate via phosphofructokinase to fructose 1,6-bisphosphate. That uses an ATP as well. It's a kinase. Okay, it's going to use ATP and it's going to use magnesium ion because if you're dealing with ATP, you better stabilize the ATP leaving. Form fructose 1,6-bisphosphate. That is a symmetric molecule. And actually, before we continue, again, remember PFK is the major flux controlling enzyme. This is where we allosterically modulate that. AMP is the big modulator. The AMP, high levels of AMP is going to increase the action of PFK2. It's going to activate PFK2. High levels of ATP is actually going to inhibit PFK. Okay. Fructose 1,6-bisphosphate is the symmetric molecule, and we can cleave that via aldolase doing a retroaldol mechanism to form GAP and DHAP. DHAP is converted to GAP via triose phosphate isomerase. Remember that goes through an ene dilate intermediate. My mnemonic is triosine phosphate isomerase. Okay, goes through an ene dilate intermediate to form that GAP. GAP can then via glyceraldehyde 3-phosphate dehydrogenase can form 1,3-bisphosphoglycerate. It formed 1,3-bisphosphoglycerate. That means you added a phosphate. You need to get that from somewhere. That's coming from inorganic phosphate. And it's a dehydrogenase, so you better be forming an NADH or a flavin compound. In this case, it's NADH. 1,3-bisphosphoglycerate via phosphoglycerate kinase to form 3-phosphoglycerate. All right, we're in our payoff phase, so you know what? We're forming ATP when we're using kinases in our payoff phase. And again, the magnesium ion because it's kinase. 3-phosphoglycerate via phosphoglycerate mutase to form 2-phosphoglycerate. Use that histidine residue to just move over the phosphates one molecule. It has a 2,3-BPG intermediate, which we mentioned. The red blood cell often gives up the opportunity to make an ATP further down the line in order to form 2,3-BPG to help us out when we have low levels of oxygen in the tissue beds. 2-phosphoglycerate via enolase and magnesium ion to form phosphoenolpyruvate, and then phosphoenolpyruvate via pyruvate kinase to form pyruvate. And that formed the ATP again as well, and it uses magnesium ion as well as potassium ion to do that. Recall that we can go through either the Tca cycle or then the glycolytic cycle, which contains pyruvate, a lactate dehydrogenase to form lactate, and that ends up getting more NAD plus, so that we can run glycolysis over and over and over. And that we do that with our fast twitch muscles. And from now we'll move into Tca cycle. Okay, intro to the citric acid cycle. The Tca cycle, also called the Krebs cycle, the tricarboxylic acid cycle, Tca cycle, has a plethora of intermediates, all directed at providing reducing equivalents, NADH and FADH2, to the electron transport chain to ultimately produce ATP. It produces that ATP by providing the electrons via these reduced equivalents to then power the electron transport chain, to transport electrons via the complexes that then protons follow and build up that gradient to then be dissipated, and while that dissipation is occurring, form ATP via the ATP synthase. The Tca cycle occurs in the matrix of the mitochondrion. Per turn of the Krebs cycle, you get 3 NADH, 1 FADH2, flavin adenine dinucleotide, 1. Per turn, you get 3 nicotinamide adenine dinucleotide, reduced nicotinamide adenine dinucleotides, 1 reduced flavin adenine dinucleotide, 3 protons, 2 carbon dioxide, and 1 GTP. Some people say ATP because GTP and ATP is easily convertible. The rest are intermediates. The rest of the compounds in there are all intermediates. Arguably, somebody can say oxaloacetate is not an intermediate, but for our purposes, that's an intermediate. Per glucose, you get double that, right? Because per glucose, we put it through glycolysis, and we end up getting 2 pyruvates to form then 2 Acetyl Co. So you get 2. Keep in mind that these intermediates that we mentioned earlier can be siphoned off into things like fatty acid, cholesterol, and amino acid synthesis, as opposed to going back into the Tca cycle. By that same token, many of the intermediates can be fed into via the aforementioned pathway. So you can fit in amino acids into the Tca cycle. You could fit in glycerol into the Tca cycle. You can fit in fatty acids into the Tca cycle. Okay. We get about 2.5 ATP per NADH molecule that we make, and we get about 1.5 ATP per FADH2 molecule that we make via oxidative phosphorylation or electron transport chain. So just a quick introduction to the players here. This is coenzyme A. Coenzyme A just has this ADP, this phosphorylated ADP group, and this chain of carbons and then amine groups, and at the end of it, it has this sulfur group that has the capability of forming thioesters, which we know can be used to power reactions because thioesters are highly reactive. So this is coenzyme A, introduction to the players. This is the new guy for you as well. This is thymine pyrophosphate, TPP. It's going to be used in pyruvate T dehydrogenase, as well as alpha ketoglutarate dehydrogenase. And it uses that E1, E2, E3 reaction mechanism that we're going to go through. And it's found in E1 rather, out of that mechanism. And what it does is it makes alpha keto acids thinks they are beta keto acid, and they spontaneously decarboxylate, and they do that via attacking the alpha keto acid. They attack the alpha carbon, the carbonyl carbon there, and they make it think that it's a beta keto acid. And the rule about that is that they spontaneously decarboxylate. So that's what it does. It gives up this proton, it becomes a carbanion, which then obviously looks much like a nitrile group or a cyanide group, and then it can attack the alpha carbonyl carbon, that partial positive charge. Alright, we already introduced you to NAD, nicotinamide adenine dinucleotide. I'm just showing you it again to understand what the reactions are occurring here. What's going to happen here is there's going to be a hydride molecule that attacks right here, and then electrons move here, electrons move here. Okay, I mean, that's what it is. It's an electron carrier, guys. It's used to then transport to the electron transport chain to push electrons through there. It's a reducing equivalent, that's what I call them. Again, another electron carrier, the flavin adenine dinucleotide, right? I mean, here's the structure. Again, what's happening here is you get this partial, the hydrogen with one electron to attack, right? And you get another semiquinone form, a radical form, and then another hydrogen with a radical electron to come in, and you get this dihydroflavonoiding dinucleotide, or FADH2. Again, not complicated. Okay, so here's where the citric acid cycle is occurring. It's occurring in the matrix of the mitochondria. So we have an outer membrane, we have the intermembrane space, we have the inner membrane, then we have the matrix of the mitochondria. Okay, and within that matrix, it has its own DNA, right? It has its own ribosomes, and within the matrix, you have the citric acid cycle occurring, providing the reducing equivalents that then go on to the electron transport chain to transfer their electrons to pump protons across these membrane proteins and ultimately create ATP by allowing the protons to go down the concentration gradient and form ATP while doing so. Using the conformational chain, the protons going down the concentration gradient, the energy from the protons going down the concentration gradient is used to cause conformational changes in this ATP synthase subunit that is transferred to this lollipop head subunit here, and then juxtaposes the ADP and inorganic phosphate to form ATP. So we'll go through this portion of it, the citric acid cycle, and then the next lecture we'll go through ATP synthase and then the electron transport chain and oxidative phosphorylation. The first step that we need to do before we get to the Tca cycle, we need to form Acetyl Co, and we do that via pyruvate dehydrogenase. And I have a mnemonic for the conversion there, and it's called pyruvate T dehydrogenase. Pyruvate T dehydrogenase, and it's not, and you don't really know what that means yet. I'm going to show you in a second. But it's important that you put the 3 E's here and not at the dehydrogenase, because they're going to be mechanisms that don't follow the same EEE mechanism, which again we'll go through it. Anyway, you take pyruvate, you take CoAsh, which is just a CoA group with a free sulfhydryl group, take NAD+, and you form an NADH and you release CO2. The mechanism which we're going to go through shortly to finally make acetyl CoA. So we're going to go through this, and we're going to go through what this EEE means. So EEE stands for E1, E2, E3. E1 is pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. The first step is to take this thymine pyrophosphate group, which again is, as I told you earlier, the cyanide-like group. It is a nitrile-like group, and what it does, it does a nucleophilic attack on this carbonyl carbon, pushing these electrons onto the oxygen, and then it picks up a proton. So it forms this hydroxyl ethyl TPP, and it forces this thing to leave, for CO2 to leave. And why it does that? Why did they do that? Well, when nitrile groups attack alpha keto acids, they make alpha keto acids think that they are beta keto acids. Pyruvate is an alpha keto acid. Clearly, this is a keto group on the alpha position. So it makes alpha keto acids think it's beta keto acids, and the rules about beta keto acids are that they spontaneously decarboxylate, so you release CO2. Okay, so now what we want to do now is we're going to take E2, we're going to take the cysteine bond from the lipoamide E2 group, and we're going to have one of these sulfur groups be attacked by this carbanion. So now what we're going to do is we're going to take our hydroxyethyl TPP, and we're going to take our E2 enzyme and take the cysteine bond from the lipoamide enzyme and have this carbanion attack one of the sulfurs, pushing the electron this way, that then goes to pick up a proton to form a sulfhydryl group. And we get acetyl dihydrolipoamide. The next step is to do a transesterification reaction with the CoA group, or the CoAsh group. Same thing. We're just going to do transesterification, so now we have the acetyl CoA group form, and now we have 2 sulfhydryl groups on our lipoamide, so we need to reform our enzyme at this point. So the next step is to use E3 to oxidize the E2 enzyme, and then we need to reform our E3 enzyme, so we take NAD+ and form NADH, and that reformed everything, and everything's good to go. We've made our acetyl CoA, and we've reformed our enzyme. Okay, so why don't we go through it broad stroke, and then we'll go through each little piece. So we start out with pyruvate via pyruvate dehydrogenase to form acetyl CoA. Of course, we have to put in a CoA group and an NAD+, because I know the mnemonic is pyruvate T dehydrogenase, use the E1, E2, E3 reaction mechanism, and so yeah, you're going to have to put in the CoA and NAD+ to get out CO2 and NADH. CO2 is coming from the fact that thymine pyrophosphate makes an alpha keto acid thinks it's a beta keto acid. Anyway, yeah, we take acetyl CoA and via citrate synthase form citrate in addition to oxaloacetate. So, and then we take citrate via convertase to form isocitrate dehydrogenase to form alpha ketoglutarate, and in doing so form an NADH and release CO2. Take alpha ketoglutarate via alpha ketoglutarate dehydrogenase, form NADH, release CO2. Succinyl CoA then we get succinyl CoA, then succinyl CoA to succinate via succinyl CoA synthetase. It's a synthetase, so we form a GTP molecule and release a CoA group. And we have to put in a pi in GDP. And now we have succinate, and now take it into fumarate via succinate dehydrogenase and form an FADH2, a reduced flavin compound, flavin editing dinucleotide compound. Take fumarate via fumarase and form L-malate, stereospecific. And then we take L-malate to form oxaloacetate, and we do that via malate dehydrogenase. And again, it's a dehydrogenase, so we form an NADH. So that's broad stroke the TCA cycle. Now I'll go through each individual part, starting with citrate synthase. Okay, reaction 1, citrate synthase. So you take acetyl CoA, some water, and some oxaloacetate. Via citrate synthase, you form citrate, and you release a CoA group. That gives free energy, standard physiological gives free energy, about negative 32.2 kilojoules per mole. It's irreversible. It's an irreversible reaction, so it's the point of regulation. As you'll come to appreciate shortly, it can be regulated by levels of NADH and oddly enough, levels of succinyl CoA. But anyway, again, it's coupled reaction. You're coupling the hydrolysis, okay, the aldol addition of methyl group of the acetate, this acetate, to the carbonyl of the oxaloacetate. And that's an unfavorable reaction, but you're coupling that with the hydrolysis of a thioester, which is the acetyl CoA group, which is a favorable reaction. So that's what allowed you to run it and give it a negative Gibbs free energy. All right, it's very, very negative Gibbs free energy. Okay. And it's actually an obligate binding mechanism. So oxaloacetate has to bind to the enzyme first in order to get this reaction to occur. And this prevents the premature hydrolysis of the thioester group, because the thioester group is coming from the acetyl CoA. This is the thioester. And you don't want that to hydrolyze before the oxaloacetate is ready to then accept the aldol addition. And so you want to prevent that, and you prevent that by having it obligate binding. Oxaloacetate has to be in that enzyme first. So here's the mechanism for citrate synthase. We're going to go through it. First, you have the formation of the nucleophile by aspartate acting as a base using base catalysis, and you're also using acid catalysis because this guy is picking up this hydrogen. So what that does now is that makes a more positive charge on this carbon, right? And that then allows for these electrons to go and flow onto right here and form this this ene group, right? Enol group. Now it can go ahead and attack the carbonyl carbon of oxaloacetate, all right? And then again do this aldol addition mechanism here. And then after that, what happens is the water molecule can then attack and hydrolyze the thioester. And now we have citrate and a release of the thioester. Again, that's what citrate synthase. Citrato synthase is the mnemonic for obligate binding. Goes through this aldol addition mechanism. All right, that was citrate synthase to form citrate. That's why it's called the citric acid cycle. So, all right, and you should know this, right? Most of you probably know this, but the 8, when you see citrate or citric acid, that's the citrate, that's the deprotonated form, and citric acid is just the protonated form. Okay, that's just carboxylic acid nomenclature. Anyway, citrate to isocitrate via aconitase. So let's go through that. I have a mnemonic, so it's called tres aconitase, really. It's an isomerization reaction. Immediately, you know that gives free energy. The approaching 0 is a reversible reaction at equilibrium. And he uses the 3-point mechanism to create the stereospecific product here, this isocitrate. And I'm going to go through that. The reality of it is, it'd be very, very difficult for you to move this guy from here to here, or from here to here, or anywhere, because of the fact that they're all the same, really. You have this CH2 connected to the carboxylic acid, this carbon connected to the carboxylic acid, the CH2 connected to the carboxylic acid. So how do you make sure to get this product? And that's what this binding mechanism is all about. What happens is that they bind here in these specific spots, and they hold them there. The enzyme holds them in these specific positions so you can get the right product out. Okay, so you get the cis aconitate intermediate, and it adds the OH group right here. Okay, so that's how you make sure that you get isocitrate and not something else. But again, it's just a hydration-dehydration reaction, right? What happens is this water molecule, you protonate this, water molecule leaves, and then something acts to the base on one of these hydrogens and pops it on there. Okay, and pops the electrons onto here to form a double bond. So then you hydrolyze it, right? So water can attack right here, and it puts the electrons right here, and then the hydrogen, and then it can pick up a hydrogen. And you have isocitrate. So that's the mechanism. All right, that was citrate via conjugate to form isocitrate. Now we're going to convert isocitrate via isocitrate dehydrogenase to alpha-ketoglutarate. And in doing so, we're going to form an NADH and we're going to release CO2. Okay, so here's the reaction. Isocitrate via isocitrate dehydrogenase from alpha-ketoglutarate. My mnemonic is bisocitrate dehydrogenase. Alright, it's an oxidative decarboxylation and it forms the beta-keto acid intermediate. The oxidation of the alcohol creates that beta-keto acid. And what are the rules about beta-keto acids? They spontaneously decarboxylate. So if you're going to form a beta-keto acid, you're going to spontaneously decarboxylate, you're going to have CO2 in the products. And that's why I put this beta here. Remember, it's the beta-keto acid intermediate, so I know I have CO2 in the products. It's a dehydrogenase, so I know I have an NADH or an FADH2 in the product. In this case, it's NADH. So here's the reaction mechanism. We're going to oxidize this alcohol group to a carbonyl group. We're going to spontaneously decarboxylate, okay, and we're just going to protonate a little bit. Protonate to get rid of this double bond here. And then we have alpha-ketoglutarate, all right, and again, bisocitrate dehydrogenase, guys, oxidative decarboxylation. Again, note the trend. Beta-keto acids are unstable and spontaneously decarboxylate. All right, we went through isocitrate. We isocitrate dehydrogenase from alpha-ketoglutarate, and we formed NADH and CO2. Now we can take alpha-ketoglutarate via alpha-ketoglutarate dehydrogenase to form NADH and CO2 and succinyl CoA. So again, here's the reaction. Okay, we take alpha-ketoglutarate to form succinyl CoA via alpha-ketoglutarate dehydrogenase, and we form an NADH, an H plus, and the CO2, and it takes a CoA group. So one thing that we should understand and know here again is this mnemonic alpha-ketoglutarate dehydrogenase. That means it uses E1, E2, E3 enzyme complex. Okay, it's the second oxidation of the cycle, second release of CO2, and it's the same mechanism as pyruvate dehydrogenase. Okay, clearly this is an irreversible reaction. You don't see a reversible arrow here. So it's an irreversible reaction. And here's the pyruvate dehydrogenase mechanism, right? Sorry, this got cut off. Here's the pyruvate dehydrogenase mechanism. Here's the alpha-ketoglutarate dehydrogenase mechanism. It's the same mechanism. It's the same mechanism. All right? What happens is you take the alpha-ketoglutarate, and I just wrote R to impress upon you that it's the same mechanism. Okay, and you decarboxylate, you release CO2 via the thymine pyrophosphate, making an alpha-keto acid think that it's a beta-keto acid. Notice the name alpha-keto. That's so you know it's an alpha-keto acid. But it makes it think it's the beta-keto acid, so it's spontaneously decarboxylate. And it does that because it's a cyanide-like group, and cyanide-like groups make alpha-keto acids think they're beta-keto acids. So this is thymine pyrophosphate. It loses proton and attacks right here, causing the CO2 to leave. All right, now we have this intermediate. And then we take E2 and it replaces thymine pyrophosphate there and forming that carbonyl carbon. And then CoA group replaces this sulfur group. So we do a transesterification reaction. Now we form succinyl CoA. And now we're just reforming the enzyme between E2 and E3. So we oxidize E2 and then E3 needs to replace itself now. So NAD plus oxidizes E3 and forms NADH. I mean, that's what it is, guys. Remember, E1 is alpha-ketoglutarate dehydrogenase, E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. All those should make sense. Transacetylase, moving the acetyl group, dehydrogenase, just oxidizing the E2. Okay, so that all makes sense. That was alpha-ketoglutarate to succinyl CoA via alpha-ketoglutarate dehydrogenase. Remember the mnemonic, alpha-ketoglutarate dehydrogenase. Remember E1, E2, E3 reaction mechanism. And because you know that an NADH is in the products. Since you know that, you know the CO2 is in the products. And you know at the end of the reaction mechanism, you're going to have a CoA type molecule. Succinyl CoA in this case. Now we're going to go through is succinyl CoA via succinyl CoA synthetase to form succinate, and in doing so we're going to form a GTP because it's a synthetase. Remember, synthetases form NTP molecules, and in doing so we're going to release a CoA group. So here we are. Succinyl CoA via succinyl CoA synthetase to form succinate. Alright, so we take pi, we take GTP, and we form a GTP molecule, and we release a CoA group. And it's the opposite of what makes sense. It's counterintuitive because you're saying succinyl CoA synthetases, synthesizing succinyl CoA, but the reality is you're taking succinyl CoA and you're forming succinate. The way I remember this is that it's the opposite of what makes sense, and I also remember the fact that when you take alpha-ketoglutarate dehydrogenase, you have to form a CoA group. So that CoA group is succinyl CoA. It's not succinate. Okay. Again, it's a coupled reaction, and as I mentioned earlier, synthetase either form or hydrolyze NTP. This one forms ETP via substrate level phosphorylation. It's a coupled reaction, and I'll show you that in a second. Keep in mind that GTP and ATP are in equilibrium with one another, and they can very easily switch between one another. The Gibbs free energy for their switching is 0, so they can switch very easily in between one another. And I should probably put more reactants in here because it's not like they just magically transform into one another, but they're in equilibrium. And I told you it's a coupled reaction, so I want to show you that. This is the first reaction. GDP plus pi to form GTP. Second reaction, succinyl CoA plus water forms succinate and CoA. You notice that the first one was non-spontaneous. It had a Gibbs free energy of 30.5. Whereas the second one is very spontaneous. It has a negative Gibbs free energy of negative 33.8. So overall, it has a Gibbs free energies. If you add up the reactions, you have to add up the Gibbs free energies. So the change in Gibbs free energy for the total reaction is negative 3.3 kilojoules per mole. Is spontaneous. I don't really show this, but you should be able to do this easily. I mean, should be child's play for you. It's what we did in general chemistry, so I don't even show these most of the time. But oftentimes you can do this with most of the reversible reactions that we go through. It's usually just coupled reactions. So that was succinyl CoA to succinate via succinyl CoA synthetase, and when we did that, we formed a GTP via GTP and inorganic phosphate, and we released a CoA group. I didn't want to show you the mechanism. It's not that serious. Let's not go there. Let's not get complicated. And we take succinate via succinate dehydrogenase to form fumarate. So the big thing about succinate dehydrogenase is that it's not forming an NADH molecule, but it's forming an FADH2, a reduced flavin adenine dinucleotide molecule. The other thing about it is that it's found in the electron transport chain. So we'll get there right now. Again, succinate via succinate dehydrogenase to form fumarate, and that forms then a reduced flavin adenine dinucleotide molecule. Okay. It's a simple redox reaction, and it's the enzyme found in complex 2 of the electron transport chain. Here's complex 2, right here. So it's found right here. So this reaction occurs right here. Succinate to fumarate, all right? And that then provides electrons that go to then ubiquinone, and then travel to complex 3, it forms hydroquinone, a hydroquinone, and then it travels to complex 3 to give an electron there, go through the Q cycle. Those electrons eventually get to cytochrome C. Cytochrome C transfers the electron to complex 4. Complex 4 eventually transfers the electrons to the oxygen to form water. And all through doing so are pumping these protons that then can then dissipate the gradient and go through the ATP synthase, go through this direction right here, to then form ATP. Right, the conformational changes associated with the protons going down their concentration gradient are transferred to this area right here, and they undergo a conformational change that juxtaposes the ADP and inorganic phosphate to then form ATP. But anyway, the important part about here is succinate dehydrogenase is found within the integral membrane protein of complex 2. Okay, and it converts succinate to fumarate via that redox reaction. All right, so we're at succinate, and we converted succinate to fumarate via succinate dehydrogenase, and we formed a flavin adenine dinucleotide, and we know that that's going to happen because this is a dehydrogenase. Now we can take fumarate via fumarase and convert it into L-malate, a stereospecific product. It's a stereoselective reversible hydration dehydration. I almost don't even want to insult you by trying to tell you the mechanism. I mean, it's a reversible. You hydrate this and you dehydrate it. Well, you just hydrate it rather. Dehydration would be the reverse reaction. Okay, but that's the reaction. Fumarate via fumarase to L-malate. Alright, here's the mechanism. And I'm sorry to insult you, but take the hydroxyl group, you attack here, you put the carbanion here, an intermediate, and then the proton goes and attaches right there. And you form L-malate. Stereospecific. The enzyme is just built to only make L-malate. Okay, and then the last step here is L-malate to oxaloacetate via malate dehydrogenase, and in doing so you form that NADH molecule again because it's a dehydrogenase. It's a redox reaction. It's the formation of a beta keto acid. So oxaloacetate is the beta keto acid. Okay, the rules about beta keto acids are that they are unstable, they decarboxylate. So you need to use this oxaloacetate very quickly. And one of the good things is that in the obligate binding phase of citrate synthase, oxaloacetate binds first. So that's very good. All right. and it pulled it forward. Okay, it pulled it forward. The reaction for citrate synthase is a very negative reaction. It's a very spontaneous reaction. It has a Gibbs free energy of about, I don't know, like -40 standard physiological Gibbs free energy conditions. So it pulled that reaction forward very, very hardly, very, a lot. So as soon as you form oxaloacetate, it doesn't decarboxylate because it's pulled into the reaction with citrate synthase to form then citrate. But anyway, this is L-malate via malate dehydrogenase form oxaloacetate, and you form an NADH and a proton there. And that's the citric acid cycle. So we can go through it again, and I'll go through a broad stroke now. This isn't part of the cycle per se, but you take pyruvate via pyruvate T dehydrogenase to E1, E2, E3 reaction mechanism, and you form Acetyl Co. And in doing so, you release CO2 and you form an NADH. E1 is the thymine pyrophosphate. TPP attacks a carbonyl carbon, making an alpha keto acid. Think it's a beta keto acid. Spontaneous decarboxylation occurs. And then you attack via the E2 group, the dihydrolipoylamide group. All right. And you form this thioester group that then can be replaced by the CoA ester group by a transesterification reaction, and then you just re-reduce the enzyme E2 via E3 and NADH. NAD plus oxidizes the compound, the enzyme, back to its normal form and reduces itself. Okay. We take acetyl CoA and via citrate synthase with oxaloacetate to form citrate. What you need to remember about that is that it's obligate binding and oxaloacetate binds first. Okay, we form citrate, and it pulled the reaction forward via mass action. And you notice there's no reversible arrows here. It's a very exothermic, endo-exergonic reaction, rather. It gives free energy very low. We take citrate via aconitase to form isocitrate. What you need to remember about that is that 3-point binding mechanism. So tres aconitase, that's 3 in Spanish. Tres aconitase to form that stereospecific product, isocitrate. Then we take isocitrate dehydrogenase to form alpha ketoglutarate. And the mnemonic for that is bisocitrate dehydrogenase. It forms a beta keto acid intermediate. Well, the rules about beta keto acid is that they spontaneously decarboxylate. They're going to release CO2. And you're going to form an NADH because it's a dehydrogenase. Okay? So that's what you need to remember about that. Alpha ketoglutarate via succinyl CoA via alpha ketoglutarate dehydrogenase. Again, remember the mnemonic alpha ketoglutarate dehydrogenase. All right, you're going to release CO2, you're going to form an NADH, and you're going to form a CoA product. And that's just from the fact that the E1, E2, E3 reaction mechanism is built like that. So you form succinyl CoA, you form an NADH, you form a CO2. Take succinyl CoA via succinyl CoA synthetase to form succinate. In doing so, you form a GTP. How do I know that? How do I know you form a GTP? It's a synthetase. Synthetases form NTP molecules or break them down. And you have to put in the inorganic phosphate to do that, because you're not getting phosphates anyway. There's no phosphates in this molecule. And then we can take succinate via succinate dehydrogenase to form fumarate. Things you got to remember about that is you're forming a reduced flavin adenine dinucleotide compound, as opposed to then a nicotinamide adenine dinucleotide compound. The other thing is that it's found in the electron transport chain in complex 2. Okay, it's part of that complex. All right, and that's a big step in getting electrons to ubiquinone, that Q molecule. Take fumarate that results from that via fumarase to form L-malate. All right, not much to say about that except the fact that it formed the stereospecific product, and then L-malate via malate dehydrogenase to form oxaloacetate, and you form an NADH there because it is a dehydrogenase. Again, oxaloacetate is a beta keto acid, but because it's unstable, but it gets pulled so quickly into the formation of citrate that it's okay. It's not going to hurt us too much. It's not going to go nowhere. Okay. The Tca cycle, also called the Krebs cycle. So net products, right? How many net products? We'll go through that in the next page. The Tca cycle, also called the Krebs cycle or the tricarboxylic acid cycle, is a plethora of intermediates, all directed at providing reducing equivalents, NADH and FADH2, to the electron transport chain to ultimately produce ATP. The Tca cycle occurs in the matrix of the mitochondrion. Per turn, you get 3 NADH, 1 FADH2, 3 protons, 2 CO2, and 1 GTP. The rest are intermediates. Some people call this an ATP because they're in equilibrium with one another. Per glucose, you get two times that, right? Because you're getting per glucose, you get 2 pyruvates for every glycolysis reaction you run, or 2 acetyl-CoAs, which way you want to look at it. All right, keep in mind that the intermediates that were mentioned can be siphoned off into things like fatty acid, cholesterol, amino acid synthesis, so you don't always get these products, but by the same token, many of these intermediates can be fed into via the aforementioned pathway, so you can get amino acids feeding into the Krebs cycle. You can get fatty acids feeding into the Krebs cycle via acetyl-CoA. So, you can go both ways. We get about 2.5 ATP per NADH and 1.5 ATP via the subsequent pathway oxidative phosphorylation, okay, which we'll go through next. Probably not in this lecture, but next.